Hi, we're Team Blood from the Gemstone Honors Research Program here at the University of Maryland, and we need your help in funding our project. Acute myeloid leukemia is a type of cancer that affects approximately 20,000 people annually in the United States. AML accounts for 25% of all leukemias in adults and has the lowest survival rate. Typically, bone marrow contains hematopoietic stem cells, which give rise to the three blood cell components: red blood cells, white blood cells, and platelets. However, in AML, the bone marrow produces immature abnormal cells that crowd out the healthy cells that the body needs. Current treatments for AML include bone marrow transplants, in which bone marrow containing healthy hematopoietic stem cells is transplanted from a donor to the patient. However, this introduces the possibility of graft versus host disease, in which there is an immunological mismatch between the donor and the recipient's body rejects the transplant. Theoretically, we could bypass this problem if you could use the patient's own cells. Recently, researchers have discovered ways to take specialized somatic cells, such as skin cells or blood cells, and reprogram them into induced pluripotent stem cells, or iPSCs, which can be intentionally differentiated into many cell types. This means that, in treating AML, we can potentially take a patient's own cells, reprogram them into iPSCs, and then differentiate them into healthy hematopoietic stem cells that will match the patient's immune system. However, it is still unclear why iPSCs differentiate, and the efficiency is still very low. Team Blood is investigating how to optimize the procedure of differentiating iPSCs into hematopoietic stem cells by analyzing the proteins that facilitate this process. iPSCs can differentiate into HSCs through co-culture with a type of stromal cell known as OP9 cells. When co-cultured, these cells secrete proteins that drive this differentiation. Our aim is to determine whether the OP9 cells themselves are necessary to induce a differentiation, or if simply the proteins secreted by the cells are enough. By culturing iPSCs with direct and indirect contact with OP9 cells, we can identify which elements in the OP9 cells are crucial for the differentiation of iPSCs into HSCs. Our research project seeks to isolate and identify the proteins that are responsible for differentiation to occur. We wish to optimize the methodology, understand the mechanism behind differentiation, and ultimately obtain a greater yield of HSCs that can be engrafted into leukemic patients. Team Blood is in need of your help, and we would greatly appreciate any and all donations you are able to give. Thank you.